So the chapter 10, that is biotechnology and its application. So context we are going to study is the biotechnological application in agriculture, biotechnologically applications in medicine, transgenic animals and ethical issues. Okay, so biotechnology essentially deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. So, the application of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, wastewater, and energy production. So, three critical research areas of biotechnology are okay. Providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Creating optimal conditions through engineering for a catalyst to act. Okay. Now downstream processing technologies to purify the protein organic compound. So these are the three critical research areas of biotechnology. Okay. So biotechnology to improve the quality of human life, right? Especially in the field of food production and health. Especially in the field of food production and health. So biotechnological applications in agriculture. Okay. For increasing food production, right? We know the agrochemical based agriculture, organic agriculture, and genetically engineered crop based agriculture. So, now the Green Revolution succeeded in tripling the food supply, but it was not enough. Okay, increased yields have partly been due to the use of improved crop varieties, but mainly due to the use of partly have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals, that is fertilizers and pesticides. Okay, that is fertilizers and pesticides, right? Now how fertilizers and pesticides, right? However, for farmers in the developing world, agrochemicals are often expensive. Okay, so not possible using the conventional breeding. So as traditional breeding techniques fail to keep pace with demand and provide sufficiently fast and efficient systems for crop improvement, another technology called tissue culture got developed. Okay, now what is tissue culture? In during 1950s, that whole plants could be regenerated from explants, that is any part of the plant taken out and grown in a test tube under sterile conditions in special nutrient medium. Now this capacity to generate a whole plant from any cell or explant is called totipotency. This capacity to generate a whole plant from any cell explant is called totipotency. Okay. Now the nutrient medium must provide a carbon source such as sucrose and also inorganic salts, vitamins, then amino acids and growth regulators like auxins, cytokinins, etc. So by this we can propagate of large number of plants in very short duration. Now this method of producing thousands of plants through tissue culture is called micropropagation. So okay, this is called micropropagation. Now each of these plants will be genetically identical to the original plant from which they were grown, that is they are soma clones, right? So many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. So these are the micropropagations, and then genetically identical to the original plant from which they were grown, that is they are soma clones, right? Another important application of the method is the recovery of the healthy plants from diseased plants. Even if the plant is infected with a virus, the meristem, that is apical axillary, is free of virus. Hence one can remove the meristem and grow it uh, vitro in vitro to obtain virus free plants. Scientists have succeeded in culturing meristems of banana, sugarcane and potato. Now scientists have even isolated single cells from plants, and after digesting their cell walls have been able to isolate naked protoplasm surrounded by plasma membrane, right? After digesting their cell walls have been able to isolate naked protoplasm surrounded by plasma membrane. Isolated protoplasts from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplasts which can further grown to form a new plant. These hybrids are called the somatic hybrids, while the process is called somatic hybridization. So in somatic hybridization, naked protoplasts are fused to get the hybrid protoplast. Okay, and in meristem, in micropropagation, they have the somaclones, micropropagation, and protopatency, uh, regeneration of the whole plant from a explants, that is any part of the plant taken out and taken in a test tube under the sterile condition in a special nutrient medium, right? Now, is there any alternative now? Okay, so imagine a situation where protoplast of tomato is fused with the with potato. Now they are grown, so tomato plus potato is pomato, but to from new hybrid plants combining tomato and potato. But unfortunately, the plant did not have all the desirable combination. Okay, now any alternatives for this, like because is there any way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crops is a possible solution. So plants, bacteria, fungi, animals whose genes have been altered by manipulation are called genetically modified organisms, that is GMO. Plants, bacteria, fungi and animals whose genes have been altered by manipulation are called genetically modified organisms, GMO. Now genetically modified plants have been useful in many ways. That genetic modification has made crops more tolerant to abiotic stresses, that is cold, drought, salt and heat. Made crops more tolerant to abiotic stresses, that is cold, drought, salt and heat. Reduced reliance on chemical pesticides, that is pest resistant crops. Okay, now help to reduce post harvest losses, increase efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil. Increase efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil. Enhance nutritional value of food, that is golden rice, that is vitamin A enriched rice. So GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels and pharmaceuticals. Okay, are the production of pest resistant plants which could decrease the amount of pesticide use. Okay, pest resistance plant. Bt toxin is produced by a bacteria called Bacillus thuringiensis, Bt for short. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticides. In effect created a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato and soybean. So Bt cotton. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans, tobacco, budworm, armyworms, coleopterans, that is beetles, diphterans, flies and mosquito. Okay, some strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans, that is tobacco, budworm and armyworm, and coleopterans, that is beetles, then diphtheria, that is flies and mosquitoes. So Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contain a toxic, that is insecticidal protein. Why does this toxin not kill the bacillus? Because actually the bt toxin protein exists as inactive prototoxins, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. Okay, the activated toxin binds to the surface of mid gut epithelial cells and create pores that cause cell swelling and lysis and eventually cause the death of the insects. So due to the alkaline pH of the gut, we solubilize the crystals, and then activated toxin binds to the surface of the mid gut epithelial cells. Right now, inactive prototoxin, right, and insecticidal protein. These crystals. Now these crystals contain the crystals inside contain the insecticidal protein in the form that is inactive. The Bt toxin protein exists as inactive prototoxin, but once ingested digest, they first solubilizes the crystals, and then now the activated toxin binds to the midgut. Okay, so specific Bt toxins genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. The choice of genes depends upon the crop and the targeted pest. Okay, As most BT toxins are insect group specific, the toxin is coded by a gene cry1AC and named cry. The toxin is coded by a gene cry1AC named cry. The toxin is coded by a gene cry1AC named cry. The gene is cry. There are number of them cry genes. For example, the proteins encoded by the genes cry1AC. So main gene is the cry. Now they have the number of genes like the encoded by the genes cry1AC, cry2AB control the cotton bollworms, and cry1AB controls the corn borer. So cry1AC and cry2AB control the cotton bollworms, and cry1AB controls the corn borer. Cry1AB controls the corn borer. Cry1AC and cry2AB controls the cotton bollworms, and cry1AB controls the corn borer. Right now, cotton ball you can see destroyed by bollworms in the first diagram. Then a fully mature cotton ball. Okay, so pest resistant plants. Several nematodes parasitize a wide variety of plants and animals, including human beings. A nematode Melodico incognita infects the roots of the tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference, that is RNAi. Now, RNAi, that is RNA interference, takes place in all eukaryotic organisms as a method of cellular defense. Now, this method involves silencing of a specific mRNA due to a complementary double-stranded molecule that binds to and prevents translation of the mRNA, that is silencing of the mRNA. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements transposons that replicate via an RNA intermediate. Now, using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double standard RNA that initiated RNA interference and thus silenced the mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific RNA. The transgenic plant therefore got itself protected from the parasite. Right now, the host plant generated double standard RNA triggers protection against nematode infestation. You see now the first diagram, roots of a typical control plants, and second is the transgenic plant roots 5 days after deliberate infection of nematode but protected through novel mechanism. Now, biotechnological applications in medicine. So the recombinant DNA technological process have made immense impact in the area of healthcare by enabling mass production of the safe and more effective therapeutic drugs. Okay. Further, the recombinant therapeutics do not induce unwanted immunological responses as in common in case of similar products isolated from non-human resources. In case of similar products isolated from non-human resources, at present about 30 recombinant therapeutics have been approved for human use. The the the, the world. Okay, in case of similar products isolated from non-human sources, so at present about 30 recommended therapeutics have been approved for human use in the world over. In India, 12 of these are presently being marketed. Okay, now genetically engineered insulin. So management of adult onset diabetes is possible by taking insulin at regular time intervals. What would a diabetic patient do if enough human insulin was not available? If enough insulin was not available. Okay, if you discuss this, like use insulin from other animals, that could be an alternative, but would the insulin isolated from the other animals be just as effective as that secreted by the human body itself, and would it not elicit an immune response in the human body? Now imagine if bacteria were able that could make human insulin. Now suddenly the whole process becomes so simple. You can simply grow a large quantity of the bacteria and make as much as insulin as you need. But think about whether insulin can be orally administered to diabetic people or not. Why? Because in stomach, protease can digest the protein, a peptide, which is insulin. So this is why it cannot taken original or orally, it is through the blood. That's the best way. Now, insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Now, from pancreas of slaughtered cattle and pigs. Now, insulin from an animal source, though, cause some patients to develop allergy. It causes allergy or other types of reactions to the foreign protein. Now, insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also needs to be processed before it becomes a fully matured and functional hormone, which contains an extra stage called the C peptide. This C peptide is not present in the mature insulin and is removed during maturation into insulin. Now, the main challenge for production of insulin using rDNA technique was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains, and introduced them in plasmids of E. coli to produce insulin chains. Chains A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. Okay, so maturation of proinsulin into insulin, as you can see, the removal of the C peptide bond, that is pre-peptide bond. A has one disulfide bonds, and between A and B has two disulfide bonds, S and S. Proinsulin has three disulfide bond. Okay, so now coming to the point, that is gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Now, gene therapy is a collection of methods that allows creation of correction of a gene defect that has been diagnosed in a child embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Now, correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. Correction of a genetic defect involves delivery of a normal gene. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. Okay. um Now, the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency, that is ADA deficiency. Now, this enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. Okay, but the problem with both of these approaches that they are not completely curative that they are not completely curative, right? They are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. As a first step towards gene therapy as a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. The functional ADA-cDNA using a retroviral vector. It was a retroviral vector, okay, not E. coli. It was a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolate from marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. Okay, so this is the gene therapy. Now coming to the point, that is molecular diagnosis. So you know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis, serum and urine analysis, early detection is not possible. So recombinant DNA technology, polymerase chain reaction, and enzyme linked immunosorbate assay are some of the techniques that serve the purpose of early diagnosis. Now, presence of a pathogen, bacteria, virus is normally suspected only when the pathogen has produced a disease symptom. Presence of pathogen is normally suspected only when the pathogen has produced a presence of pathogen bacteria viruses is normally suspected only when the pathogen has produce a disease symptom, but by this time the concentration of pathogen is already very high in the body and the person may gets weak. now however now however now however very low concentration of a bacteria or viruses can be detected at when the symptoms of the disease are not yet visible, it just started, but we can just detect it by how? By amplification of the nucleic acid by PCR. Now can you explain how PCR can detect very low amount of DNA? PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutations in genes in suspected cancer patients too. So cancer and AIDS, it has the application. It is powerful technique to identify many other genetic disorders, and even genetic disorders also solved by the PCR. So this is the polymerase chain reaction. A single standard DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection in a clone of cells followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film. The clone having the mutated gene the clone having the mutated gene the clone having the mutated gene will the will hence not appear on the clone having the will hence not appear on the photogrammic, because the probe will not have complementary with the mutated. If the clone having the mutated gene will hence not appear on the photographic film, because the probe will not have complementarity with the mutated gene. Only genes will be shown that has the complementary with the probe, but as there is mutated gene, that is extra gene, that is unknown gene, so the probe does not have the complementarity with it. So this is why it does not show. Now coming to the point that ELISA enzyme linked immunosorbate assay is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens, that is proteins, glycoproteins, or by detecting the antibody synthesis against the pathogen. So the ELISA is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens, that is, and now antigens can be protein antigens, like the antigens can be proteins, glycoproteins, or by detecting the antibody synthesis against the pathogen, or the antibody synthesis against the pathogen. Now 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 the transgenic animals. Animals that have had their DNA manipulated to possess. So the transgenic animals, the animals that have had their DNA manipulated to possess and express an extra foreign gene, are known as the transgenic animals. So transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. Although over 95% of all existing transgenic animals are mice. 95% of all existing transgenic animals are mice. Why are these animals being produced? How can man benefit from such modifications? So it is used to study the normal physiology and development. So transgenic animals can be superficially designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. The study of complex factors involved in growth, such as insulin like growth factors. A study of complex factors involved in growth, such as insulin like growth factors. Now the study of diseases. So many transgenic animals are designed to increase our understanding of how genes contribute to the development of diseases. Right? Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of diseases. Now today transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. So cancer, cystic fibrosis, rheumatoid arthritis, which is an autoimmune disease, and then Alzheimer's, right? Third is the biological products. Now medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA or genes which codes for a particular product, such as human protein, that is alpha 1 antitrypsin, used to treat epicema. Similar attempts are being made for treatment of phenylketonuria, that is PKU, and cystic fibrosis. So epicema, then phenylketonuria, and then cystic fibrosis. So in 1997, and Eli Lilly in 1983, right? So in 1997, the first transgenic cow, that is Rosie, it is transgenic cow, not buffalo, it is Rosie, produce human protein enriched milk. Produce human protein enriched milk, that is 2.4 grams per liter. The milk contains the human alpha lactalbumin. This milk contains an human alpha lactalbumin, was nutritionally a more balanced product for human babies than natural cow milk. Okay, because it contains human alpha lactalbumin. Now the vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used. Before they are used before they are used on humans. So transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of the monkeys to test the safety of the batches of the vaccine. First they use the transgenic mice, and then they can be replaced with the monkeys. Chemical safety testing. Now this is known as toxicity safety testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes now which make them more sensitive. Let's take an example. If I introduce lots of genes into your body, then your body will be like sensitive to anything, like the same things will like that. If the transgenic animals we just put that carry genes, lot of genes which make them more sensitive to toxic substances than non-transgenic animals. Now these sensitive transgenic animals, now they are then exposed to the toxic substances and the studies are affected. So this is how drugs and safety gets checked. So toxicity testing in such animals will allow us to obtain the results in less time. So ethical issues now. So biotechnology has some ethical issues as well. Now, the manipulation of living organisms by the human race cannot go on any further without regulation, because some ethical standards are required to evaluate the mortality of all the human activities that might help or harm the living organisms. Going beyond the mortality of such issues, the biological significance of such things is also important. Going beyond the morality of such issues, the biological significance of such things is also important. It should be a biological significance, right? Genetic modification of organisms can have unpredictable results when organisms are introduced into the ecosystem. Therefore the Indian genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Now therefore the Indian government has set up the organizations such as GEAC, the Genetic Engineering Approval Community, appraisal or approval community, which will make the decision regarding the validity of GM research and the safety of the introducing GM organisms for public services. So the validity of GM research and the safety of the introducing GM organisms for public services. Now the modification usage of living organisms for public services as food and medicine sources, for example, has also created problems with patents granted for the same. The modification usage of living organisms for public services such as food, medicine sources, for example, has also created problems with patents granted for the same. There is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic materials, plants, other biological resources that have long been identified, developed, and used by the farmers and indigenous people of a specific region, country. So this is the public anger. Okay, they grant the patents for products. It's kind of stealing. If you rob something, it's that kind of thing, right? Without asking for, or just stealing something. Rice is an important food grain, the presence of which goes back thousands of years in Asia's agriculture history. So rice, the biggest food in Asia's agriculture history, that is rice, that is main food grain. There are an estimated 2 lakh varieties of rice in India alone. 2 lakh varieties of rice alone found in India. 2 lakh varieties of rice. We eat, we might know 1, 2, or 3. That's all. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique flavor and aroma, and 27 documented varieties of basmati are grown in India. So there is reference to basmati in ancient texts. False. Okay, poetry. It is not that important. Now, in 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed with a semi-dark varieties and claimed as an invention or a novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent uses like products and processes based on the Indian traditional herbal medicines, that is turmeric and neem. So turmeric, neem and basmati being patent. So these three things has been patented by the people, by the different countries, right? Now turmeric and neem. So biopiracy is the term used to refer to the use of bioresources by multinational. So biopiracy is the term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. I read it again, like biopiracy is the term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Okay, so most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing country, the developing like the India and the underdeveloped, which is rich in biodiversity and traditional knowledge related to bioresources. So traditional knowledge related to bioresources can be exploited to develop modern application and can also be used to save time, effort, expenditure during their commercialization. Not very important, the last line, but the starting two lines are important. There has been growing realization of the injustice, inadequate compensation and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. There has been growing realization of the injustice, inadequate compensation and benefit sharing between the developed and developing countries. Therefore, some nations. So to make not happen this injustice, now some nations are developing laws to prevent such unauthorized exploitation, like stealing someone's work, right, which they have been hardworking all days and night. Unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patents Bill that takes such issues into consideration. Now the Indian Parliament has recently cleared the second amendment of the Indian Patent Bill. So they have made the law like Indian Patent Bill. It's passed. That takes the issues into consideration, including patents, terms of emergency provisions, research and developing initiatives. Emergency provisions, research and developing initiatives. So this is your chapter. It was it. So that's it. If anything is left, let me see. So this is a pro insulin, and then there is a mature insulin, and so yeah, this is it. Okay, we have studied like biotechnological applications in agriculture, biotechnological application in medicine, transgenic animals, and ethical issues. So all we have studied this in chapter 10, that is biotechnology and its application. Okay, so this is it.